So the different types of energy that we're looking at, we can see all of the energy can be converted between different energy types. So we get radiant energy from the sun. We have electrical energy, which of course we deal with, in the day-to-day. We see this in our, in the computers that we're using right now to look, or phones that we're using to look at this. We see chemical energy, and a lot of what we're going to be dealing with is actually chemical energy because that's the energy that's stored in the chemical bonds in every chemical, and breaking those bonds apart is how we get the energy which we convert into everything we need. There's sound energy, so the vibrations in the air, thermal energy and heat, nuclear energy, and magnetic energy, which we're not going to talk about much in this class, and then mechanical energy, which is the kinetic and potential energy that we deal with in terms of movement. So kinetic energy is the active movement right now. Potential energy is the potential that something has to fall due to gravity. So right now I'm holding this little squishy rocket up in the air. When I let it go, it drops. Here it has potential energy. Here that converts to kinetic energy. So energy is constantly being converted between these different states. And so here we're looking at an example of a monkey moving around. So we start off with the sun producing radiant energy, and the plants can convert that radiant energy into carbohydrates. So those carbohydrates are produced into the fruit. In the banana, in this case, the monkey comes in, eats the banana. That chemical energy that's stored in the carbohydrates is now stored in glucose in the monkey. And that glucose is taken to the monkey cell, which the muscle cell can now convert that glucose into ATP, another form of chemical energy, and then use that ATP to produce kinetic energy in its muscles, to contract the muscles. When that happens, some energy is going to be lost as heat to the surrounding area. Every time there's a conversion process, some energy is lost as heat. So when we look at chemical energy, we're looking at the bonds between the chemicals, and that's storing a certain amount of energy. So every chemical has a certain base level of energy stored in it, as long as it has any bonds at all. Now, we can have different amounts of energy stored in different chemicals, depending on the type of bonds they have, how stable they are, all sorts of things. Now, a reaction is classified as endergonic or exergonic based on which has more energy, the reactants, the starting place, or the products, the ending place. If it's endergonic, that means energy is going in to the product, so that endo, inside. If it's exergonic, energy is exiting the reactants. And this is important because if it's an endergonic reaction, energy has to come from somewhere. And we'll look at how that happens, but the energy needs to come from somewhere to go into that product. If it's exergonic, energy is released, and potentially we can harvest that energy and use it. To make the endergonic things we need, we often pair it with an exergonic reaction that's going to produce the energy that we need for the endergonic reaction. And for these chemical reactions, we can have chemical reactions where we take two things and put them together, where we take one thing and break it apart, where we replace one thing with another thing, or we do a double swap, where we take A and B and C and D and switch it to A and C and B and D. Now, for all of these, basically chemicals are floating around in the environment, and they're moving around by diffusion, which we talked about last semester. The higher the temperature is, the faster they're moving, and the more they bump into each other. And for a reaction to happen, all the reactants have to be in the same place and bump into each other, unless there's just one thing splitting apart. This means that at higher temperature, more reactions are going to happen because things are just going to bump into each other more often. Also, higher concentration means things are going to bump into each other more often, and reaction is going to increase. However, for most of the reactions we need in our body, this isn't actually enough. So that means that for this to happen, it doesn't happen fast enough at the temperatures we need. And so we use catalysts. And those catalysts are enzymes, which are generally proteins. There are a few exceptions. We have these enzymes, and these enzymes make it easier for the reactions to happen. Now, what's important here is they do not change the energy state of the substrate or the product. They just make it easier for it to happen. And that's because there's always a transition state in the middle that has a higher energy than either the substrate or the product. If you think about having something that you're trying to, you're putting together a piece of IKEA furniture or something, and you need to bend something into a slightly weird angle to get it to attach. But once it's attached, you can release it. It's all about getting those angles, getting that connection, disconnection, making these things happen. And so to get over that transition state, the enzyme is basically just a thing that puts everything in the right positions. And part of this is also just making sure things get to the spaces they need. So let's say you've got two things that need to react, and they're just floating around in this diffusion, random movement. And when are they going to bump into each other? Well, an enzyme grabs them and makes sure that they bump into each other in the right way. So, all right, I added another picture I don't have because of the year change. So it's just that it holds them together, pushes them together, pulls them apart, provides a little bit of force to make sure in the right angles. But it isn't changed by this. It's more like, you can think of an enzyme kind of like a screwdriver, right? It's a lot easier to screw in a screw with a screwdriver, but the screwdriver isn't affected by the process. It's just a tool that makes it easier to make this change happen. Now, for this to work, the reaction has to be one that's going to happen basically anyway. So that means, I'm not going to set up a poll for this, but these endergonic reactions that take energy in, an enzyme doesn't actually affect. It has to be exergonic. It has to have that energy change, that energy decrease. However, one thing that enzymes can do is have pieces where they pair an exergonic reaction with an endergonic reaction, so the energy can go from the exergonic reaction in. The most common example of this is breaking ATP down to ADP. ATP is our energy currency, and ATP is the lower energy form that is produced here. So we have 3 phosphates, 2 phosphates. So breaking ATP down into ADP and an extra phosphate, which makes it into a lower energy state. So if you have an enzyme that over here breaks down ATP and then uses that energy to fuel an endergonic reaction, that's how we produce things that are endergonic reactions. For example, clipping amino acids together, we're producing a higher energy state. And to do that, we need the energy to come from somewhere. All right, any questions on endergonic and exergonic reactions or on enzymes in general? We're going to do an activity on enzymes, so we'll talk about them in a lot more detail. All right, so I want to talk a little bit about the key compounds that are involved in the human body. Now, we talked a little bit about water last time. So water is H2O. Water is important for basically everything in the human body. It's involved in a whole lot of reactions, but besides being involved in reactions, it's the carrying substance in your body. It's what your blood is made out of, it's what your tissues are made out of, it's what your interstitial fluid is made out of. So most of you is water. And having enough water, it's important for carrying things around, dissolved things, for excreting waste in your urine, and also by getting rid of the waste in feces. It's important for protecting organs. We see these like the arachnoid matter in your brain, where we've got that water cushion. We use it for regulating body tissue, for lubricating joints. So water is fundamental to everything your body does. And a big part of that is acting as this solvent. Now, things can be dissolved in water to greater or lesser degrees. A true solution is when we have polar or ionic things that are small and they fully dissolve. We can also have a colloidal solution. Colloidal solution is when you have large things, but they're polar. And so they're dissolved, but they're too big for just to kind of get lost in the water. If you think about Jello, Jello is a colloidal solution. We have these large proteins in Jello, and they get sort of stuck. So we've got this texture. And that's actually really important for making up sort of the gel substance of your body, is things like that. And then there are suspensions, particles that are too large to even dissolve like a colloid. And so milk, for example, is a suspension. These are too large, or they're not sufficiently hydrophilic. And so they're floating around in the liquid, but they're not really dissolved. They will sift out if you leave the liquid. So blood, all the blood cells in your blood, that's a suspension. Any questions on the role of water there? If you have questions, by the way, when I call out for them, you can either just speak up or you can type them in the chat, whatever's easier for you. All right, so dehydration synthesis. So this is our primary method of building larger molecules, which we started looking at this a little bit in the POGL that we started working on. So to build these larger molecules, the monomers have an H on one side and an OH on the other side. And an H and an OH, it is essentially always exergonic to produce a water there. So we can produce a water here, break that down, take the water, and then this allows us to link these remaining pieces together. So we can take the water out and link this together. That's dehydration synthesis. Now, hydrolysis, we can do the other way around. It's just the opposite here, where we're putting a water back in, breaking it into H and OH. So that's our hydrolysis or dehydration synthesis. Hydrolysis breaks it down. Dehydration synthesis puts it together. Either way, we're putting in water, taking out water. In addition to that, we need to talk a little bit about pH. And we talked about pH in lab, so hopefully this should be a quick review. But we have acids and bases. So acids are things that have an H plus ion. Bases are things that have an OH minus ion. That's what we're going to talk about in biology. There are some exceptions in chemistry. With weak bases, we're not going to get into that. For our purposes, they have an OH minus ion, which means the acid and base together can always make water, bringing together that OH minus and H plus ion and neutralizing it. Now, this brings us to OH and H plus, come back to pH and POH. So pH is a measure of the acidity. POH is a measure of the alkalinity, the basicness. So acidity, and just to be confusing, pH, the lower the pH, the more acidic it is, the more H plus ions there are. For POH, the lower the POH, the more basic it is, and the more OH ions there are. Most of the time, we just talk about pH, not POH. And so a number higher than 7 means it's more basic, means that there's more OH minus ions than H plus ions. A number lower than 7 means that it's more acidic, that there's more H plus ions than OH minus ions. Now, we can, in the body, it's really important to maintain a very, very stable pH. We are only healthy if our blood pH is between 7.35 and 7.45. Any higher or lower than that, and it starts causing problems for us. And so we'll talk a lot more about that later in the semester when we get to pH management with the kidneys and the respiratory system. But here, I just want to talk about one basic way that we manage that, and that's through buffers. Now, we work with buffers in the lab, so once again, this should look familiar. This is our bicarbonate buffer system. And what happens here is this allows it to release H plus or pick up H plus ions, depending on what is happening in the body. And that causes the pH to change much less with the addition of acid or base than it would without this buffering system. Bicarbonate is our main buffering system in the body, but we have other buffering systems, including phosphate, that are constantly functioning in the body as well. The bicarbonate buffering system is really helpful because it relies on carbon dioxide and water, two things that are always available in our body. All right, any questions on pH? All right, so now we're going to come into these organic molecules that we've been talking about. And we started this with the POGL that we started on Monday, which will continue today. So all these organic molecules are based on carbon. And what's special about carbon is that it has 4 places it can make covalent bonds. So every carbon that you see in one of these atoms has 4 covalent bond locations. And these carbons then make up the backbones of these organic molecules. So in carbohydrates, we have the carb, so carbon, and then hydrate means water, which means that we've got an oxygen and hydrogens involved. So a carbohydrate is a 6-member ring, a hexagonal ring, or sometimes a 5-member ring, a pentagonal ring. But either way, that ring has mostly carbons and 1 oxygen making up the end of that ring. And that's what makes it a carbohydrate. So here we have glucose as one of our main ring, single rings, and then our monomers. And then we have sucrose here, which is a dimer, where we have our glucose. And over here, we've got our 5-member fructose. Here we've got lactose, where we have glucose, and galactose. And so these are slightly different monomers, slightly different monosaccharides that are linked together here by a dehydration reaction, right? So these are linked by that same dehydration synthesis that we talked about before. So here's our chain, and that can make starches. We also see longer complex carbohydrates, and these complex carbohydrates often produce what we refer to as fibers. Fibers are things that our bodies, our own digestive system cannot break down, but the bacteria in our gut can break down. And so they're important for sustaining our gut microbiome. And we'll talk about that a lot more when we get to digestion. Then we get to fats. So fats are hydrocarbons. What that means is that we have large areas that are just hydrogen and carbon. And this is one thing that I got some questions about, is what exactly makes a fat. And it is a little confusing because most of the fats we talk about triglycerides, that we can look at these fatty acids where they've got these long hydrocarbon tails and just a little bit of oxygen up at the top, at the end. So these are our triglycerides, and they can come in saturated and unsaturated forms. So the difference between a saturated fat and an unsaturated fat is that a saturated fat has only single bonds in those hydrocarbon tails. And so that means every carbon has a full complement of hydrogens. So there's 2 hydrogens on each linking carbon and then 3 at the end. And that means they make these nice long straight lines. An unsaturated fatty acid has one or more double bonds. And so one we call a monounsaturated fatty acid, more than one we call a polyunsaturated fatty acid. And that means that the tails go at these funny angles. Now that's important because when they all lie straight, they can pack very tightly on top of each other. When they're at funny angles, they don't pack the same way. So we get them much less tightly packed. And that's the difference as to why butter is solid at room temperature and unsaturated fats like olive oil or canola oil are liquid at room temperature. So saturated fats are solid because they're all nice, easy to pack lines. Unsaturated fats are liquid because they're funny angles. The problem is that these straight ones can also be somewhat more solid at body temperature. Now that's important for functioning for some of our cells. But if you accumulate too much of these saturated fats that are somewhat more solid at body temperature, then they can form blockages in places in your body. And this is part of why we talk about saturated fats and heart disease. All right, any questions on, so then we've got any questions on fats and saturated fats? All right, so phospholipids, which we talked about last semester in the, when we were talking about the cells, phospholipids that make up our cell membrane have a hydrophilic head, a polar head with a phosphate here, and then hydrophobic tails with these hydrocarbon lipid tails. And that's why they clump together and form that bilayer with the heads outside and the tails inside. The one other thing with these fats is there are also trans fats. People, for a while, people were using trans fats to substitute for saturated fats because we knew that there were potential issues with saturated fats. Now, trans fats Cis and trans in chemistry are just a question of what side the hydrogen ions are in a double bond. So in a cis double bond, the hydrogen ions are on the same side of the double bond. And in a trans double bond, they're on opposite sides. When they're on the same sides, you get these kinks, these bends that I was talking about in the tail. When they're on the opposite side, it goes back to being straight again, like a saturated fat. So the idea was that you could reduce saturated fats, but still have solid fats. The problem is, it turns out that these are actually even more stable at body temperature, cause more problems, accumulate more in your body. So basically, these act like saturated fat, except even more so. Turns out that these are much, much worse for you than saturated fat. Your body is pretty good at dealing with saturated fat as long as it's not a truly excessive amount. Trans fats, it does not have the same ability to deal with. Trans fats naturally exist in very small quantities in some things like butter, some natural foods, and that's okay, but it was the accumulation of large quantities of artificially produced trans fats that was really dangerous. And finally, we get to steroids. And I had a great question on the homework of someone asking, why are steroids considered a lipid? They don't look like anything else, right? They don't look like any of the other fats. And that really comes down to the fact that this basic structure is all hydrocarbon. So even though it's not a long tail, it's still all hydrocarbon. You can see there's no oxygens on here. Now, sometimes there are oxygens added on. So cholesterol has an alcohol group, that all you can hear. You can see oxygens added on in other places, but this core basic structure here is all hydrocarbons, unlike the carbohydrates, where there's an oxygen in every ring. Here, the rings are entirely made of carbon. So these steroids all come from cholesterol, which we get both through our diet and your body manufactures it. And from there, we can build all of the steroid hormones that our body needs. Cholesterol is also important structurally in producing cells. All right, any questions about anything in the fats? All right, so then that takes us to proteins. So proteins are built up of amino acids. And we've been talking about amino acid structures in the first chemical POGL that we're working on. Amino acids can be polar or non-polar, or they can even have a positive or a negative charge. And this is important because we can build proteins that are hydrophobic or hydrophilic or have hydrophobic sections. And this allows us to have proteins that can cross over the membrane. So we can have proteins that pass through the cell membrane by having non-polar sections in the middle that allow them to pass through that. So a peptide is just a series of these amino acids linked together by dehydration synthesis with this carbon-nitrogen backbone. So in carbohydrates have that carbon-oxygen ring. Peptides have this carbon-nitrogen backbone. And once we have our set of peptides, so our peptides are one amino acid chain to the next, then those peptides can form structures. And we have our secondary structures. We have alpha helices and beta sheets that form these structures that you see here. And then those come together into tertiary structures where they form a shape. And then a quaternary structure can happen where there's multiple different peptide chains that have come together to form a larger protein. And these shapes are really important to protein function. So enzymes, for example, have a particular place where the things where their products, where the reactants bind and then they take some action that moves that product, creates the product from the reactants. We also have, so for example, myosin, which we talked about a lot last semester in muscles, that needs to crawl along actin. So we have our ATP binding site. We have our actin binding region that grabs onto actin. We have regulatory chains. So we've got all these pieces that are holding it together, that are grabbing the actin, that are grabbing the ATP, all of these different structures. Myoglobin, on the other hand, is an oxygen holding protein in your muscles. And so it's globin, means glob, means it's going to be globular, round. And it's going to hold oxygen with the iron in the center right here. So whenever you hear globin, like hemoglobin or immunoglobin, you know you're looking at a sort of a round protein. All right, any questions on proteins? All right, then the last major structure that we're going to talk about is nucleic acids. So nucleic acids all have this ribose sugar here. So we've got our sugar ring here. We can see that oxygen that makes it a carbohydrate. And then it's attached to a distinct base. So here we're looking at DNA, where we have A, C, G, and T, these 4 distinct bases. So that's deoxyribonucleic acid, so ribose being the sugar, nucleic being this base here. And then we'll also have ribonucleic acid, RNA, which looks very similar. So here's DNA and RNA. So we can see deoxyribose in DNA and ribose in RNA. It's the same basic thing, except ribose has one more oxygen right here. Deoxyribose does not. So it's just that one oxygen difference. Otherwise, it's the same structure. And the big difference here is that in DNA, there's a T base. In RNA, there's a U base. Otherwise, they all act the same. Now, these bases pair up. So A pairs with C, forms hydrogen, pairs with T, forms hydrogen bonds, and G pairs with C and forms hydrogen bonds. And this gives us our structure of DNA, which allows these bonds across, forms this double helix structure that you've certainly seen before. And this one important thing that this allows, that you should hopefully remember from last semester, is the replication. By pulling these apart, we can then put a new C to bond with this G and a new G to bond with this C, and that allows us to duplicate the DNA, allowing a cell to divide and produce 2 new cells. All right, any questions on DNA and RNA? So ATP is also actually a nucleic acid. We still see this basic sugar here, our basic ribose. We still see a base coming off here, an N with an N linkage, a nitrogen linkage. But over here on this side, this is where we see these phosphate groups. So these 3 phosphate groups here are ATP. If we break one off, we get ADP. And that's an extremely exergonic reaction, produces a lot of energy. And so we can use that energy to then do anything else. So ATP is our energy currency that we use to get energy to do other things. All right, any questions on that? Okay, so we talked about the capstone a little bit on Monday, but I wanted to remind everyone about that and encourage you, when you're in your breakout rooms today, to look back at the capstone options document. Your choices for your capstone are going to be recorded in the homework for Monday, the homework that's due on Sunday for Monday. So I want you to talk today in your groups. And if you're doing a group capstone, by the end of today, you should know what type of capstone you're doing. Is it going to be a case study? Is it going to be a presentation? Is it going to be a teaching activity? And what the topic is. If you decide to do it individually, that's also totally fine. And then you should be thinking about it, but I don't necessarily need a firm decision. So if you're thinking about doing it as a group, you need to pick your type of capstone and your topic. So I'd like to just do a quick check-in on those. So what we're going to do very shortly is open up the breakout rooms, and you will finish up the activity that we started on Monday. When you finish that, so we've got stop signs on that. So the way this works on Zoom is that you should see a button that says something like call for help when you're in your breakout room. When you have finished the piece that you're currently working on, you hit a stop sign or you hit the end of the activity, you're going to hit that button. If I'm available at the time, I will come to you directly. If I am not, I will write down your group's number and then dismiss the message. Unfortunately, when I do that, it gives you some sort of kind of rude message that I have no control over that says something like, the host isn't available for you right now. But don't worry, I've written down your number and I will come to you as soon as I'm available. So I will write that down. You don't need to worry about it. Once you've gotten that dismissed, you don't need to worry about it. If it's been 10 minutes and you just want to check in, you can push it again, but I will get there. Just don't keep pushing it constantly because then it keeps showing up on my screen. So when you've finished with that, I want you to spend a little bit of time talking about the capstone. Pick your capstone. And then once you have your capstone choice, there is another activity that you can do as well, another enzymes POGL. So we're starting off with a general chemistry POGL, and we're moving into an enzymes POGL. Now, I know we started the chemistry POGL on paper. If you want to just wait and turn it on paper on Monday, that's fine. If you want to turn it in online, there's an activity for it under learning activities. The enzymes POGL is available under learning activities, and you can either turn it in. If you finish it today, that's fine. You can turn it in there. I don't expect most groups to finish it today. I expect most groups to finish it up on Monday. And so you can turn that in on Monday, either on paper or online. We'll work that out either way. Any questions about that? All right. So I do want to come back a little bit to sort of pull back on this enzymes thing that we're going to be starting. So enzymes, we have these different reaction rates that we talk about. Without enzymes, and reaction rates are pretty straightforward, right? Temperature, concentration, increase them. But enzymes get a little more complicated, where we have to look at what's going on with the enzyme. And this is important because proteins remember, those shapes are very important to how they work. And the shape of a protein can be changed by temperature or by pH, because the shape of a protein is mostly held together by a lot of hydrogen bonds. And those hydrogen bonds are going to be affected by the pH of the surrounding environment. And so that means that enzymes have particular pH ranges where they work best. So This is a POGIL activity. As I said, we started this last time, and most of you this should be familiar for, but when you're going through these POGILs, especially online, like this matters in person, but matters even more online, it's really important to make sure that you're sort of staying together. You have one person reading out loud all of these questions, and you're working together